Hello, Bio 107 students. Welcome to the Enzyme Lab. Before we start, I want to give you a little background information about what enzymes are. These are proteins, and specifically, they're proteins or chemicals that speed up a reaction without being used in the reaction. So they're often called biological catalysts because they speed up reactions for biological organisms and cells. The reactants of an enzymatically accelerated reaction are called substrates. So the enzyme is often said to be working on a substrate. Down at the bottom of the slide, we can see A plus B with an arrow over to C. And the enzyme name is often written over the arrow, although the exact writing of this can be altered. So I wanted to point that out. Sometimes we see A plus B, those would be our substrates, with the enzyme above the arrow, and C would be our product. But sometimes we see A plus B plus enzyme, arrow, C plus enzyme, or something similar to that. The important thing to note there is that the enzyme is not itself changed in the reaction. It's helping the reaction go, and the enzyme starts out as one form, and at the end of the reaction, it regains its form. So it can be used over and over again. Enzymes have a specific structure or shape and specificity. The shape is related to its protein structure. So as you remember from chapter 2, proteins have 4 levels of organization, starting out with a string of amino acids. Each individual amino acid contributes to the overall structure. The amino acids are linked together with peptide bonds, and then they will form a secondary structure. So we have our polypeptide of a string of amino acids. That's our polypeptide. Then it will either bend or spiral, forming an alpha helix or a beta pleated sheet. And then due to hydrogen bonding and interactions of the amino acids, we'll have a tertiary structure. It'll look kind of like a blob, have different shapes to it. And then when multiple polypeptides of the tertiary structure bond together, we end up with the quaternary structure and the final protein. So in this picture, each little individual circle here or shape represents an amino acid. So we can see that this is a collection of several amino acids making up this enzyme. Sorry about that. The enzyme has a specific place where the substrate will bond, and that is called the active site. The substrate here is shown in yellow, and the enzyme has wrapped around it. That's called an induced fit model or an induced fit, where the substrate triggers a change in the enzyme to make it just wrap around the substrate. At the end of the reaction, the substrate will release the product and regain its original form. So we can have one substrate going into the active site of an enzyme and coming out maybe broken apart. That would be a degradation type of reaction where the reactant or the substrate degrades into multiple products. Or we can have an anabolic reaction where we build up something bigger. And here's a picture showing an enzyme with 2 substrates going into the active site. Substrate 1 and substrate 2. They go together in the induced fit model. This is called the enzyme substrate complex, when the substrate and the enzyme are together in their specific shape. And then we end up with one product here. So here, smaller substrates have made a bigger product. This slide is showing the induced fit theory in a little more dramatic way. A is showing the active site. B1 and B2 are 2 different substrates. And in the picture over on the right of your screen, you can see how the enzyme, there we go, enzyme has changed shape, and that's temporary to allow for the bondings between B1 and B2. They will bond together and form a product. There are some factors that affect enzyme activity, and you'll see some of these in the virtual labs. Temperature can affect enzyme activity. Typically, as we increase temperature, enzyme activity will increase as well, or the rate of enzyme activity. So enzymes work faster, and they do that because as you increase temperature in a system, molecules will bounce around more quickly. They're always bouncing, but with heat, they bounce faster. And so they have more opportunities to interact and react with each other. So typically we see that increase of reactivity, increase of activity as we increase temperature. However, that can lead to a denaturation point when we have too much heat. Too much heat can permanently change the shape of an enzyme. This happens with all proteins. If you heat them up too much, and sometimes that's about 80 degrees Celsius, depending on the enzyme, it doesn't have to quite be boiling, but they can get hot enough so that they will permanently change their structure, their protein structure, and they won't work anymore as enzymes. You can think of that as cooking an egg. When you crack an egg into a pan, you can see the white as being clear, and as you increase the heat of the pan, that white permanently changes to a solid white, and it's not translucent anymore. So that's an example of denaturing a protein. We've permanently changed it. You can't uncook an egg, and the same holds true with enzymes. pH can also affect enzyme activity. Each specific enzyme has a specific pH that it works best at. So we have different types of enzymes in our bodies. We have some enzymes in our saliva. One that's very notable is called amylase, and that breaks down amylose, a carbohydrate. Our saliva has a pH of about 7. It's fairly neutral, and amylase works best at that pH. If you change the pH of your mouth for an extended period of time, the amylase enzyme activity will decrease. So we don't usually do this, even when we're eating like super sour foods or something that's very bitter. That would be more basic. But it is theoretically possible that you can alter that. Another example in our bodies is peptase. That's an enzyme that breaks down peptides or proteins in our stomach. Our stomach is very acidic. It has a pH of about 2. And peptase works great at a pH of 2. It does not work well at a neutral pH like in our saliva, and it does not work great in a very basic pH, something we might find in our small intestines. So each specific enzyme has its peak of activity at a certain pH level. Substrate concentration and enzyme concentration can both affect the rate of reaction. You're going to do a home activity where you alter the enzyme concentration. The enzymes that you'll use come from celery, and the enzyme is catalase. The more concentrated your catalase, the more activity you will have. The rate of reaction will increase. As you dilute the celery solution with the catalase, so as you dilute that enzyme concentration, you will see a decrease in enzyme activity. Same thing is true with substrates. Although we won't be testing substrates at home, you can see this done in one of the virtual labs, where if you increase the substrate in a reaction up to a point, we get an increase of reaction until we've saturated all the enzymes. So both of these work the same way, and it's shown on a chart here. Down at the bottom of this chart, we can see substrate concentration. It's increasing as we go to the right. Reaction rate is increasing on our y-axis. And we can see that as we increase the substrate concentration, we increase the reaction rate. Just as a side note, on this graph, substrate concentration is an independent variable, and the reaction rate is a dependent variable, something we're measuring as an outcome. So as we increase the substrates, shown as those little blue things, increase them with this about the same ratio of enzymes, we can speed up the reaction until all the substrates are saturating all the enzymes, and then we can't go faster anymore. So there's like a maximum level. To get that to go any faster, if we wanted to break that barrier, we would need to add some more enzymes, and then we can have workers for all those substrates. That's how I like to think of it. The enzymes are kind of like workers on an assembly line or a conveyor belt, and the substrates are coming along. When you fill them all up, fill up all those enzymes, they don't go any faster. So like I said, you will be doing some virtual labs for this lab. Those are on the McGraw-Hill site, and there you'll get to experiment with some different factors and variables. You'll also do a kitchen lab where you are measuring the rate of reactivity for the enzyme catalase, which is found in a variety of vegetables. Catalase is in potatoes, and if you take the in-person lab at HCC, or if you've done that in the past, you've used potatoes for this, but we found celery works pretty well. So we'll be using celery as our catalase. Catalase is in the celery. That's going to be our enzyme, and the substrate we're working on is hydrogen peroxide. In the lab, I say to have a bottle of hydrogen peroxide, but you're only going to need a very small amount. So if your bottle is partially used, that will be just fine. The catalase enzyme breaks apart the hydrogen peroxide to form the products of water and oxygen. The oxygen forms bubbles, and you're going to see bubbles on a little paper disc that has been soaked in the enzyme solution and dropped into the substrate solution. As the bubbles increase, the oxygen increases, that little paper disc is going to rise up, and you're going to time this. You'll be diluting the enzyme solution and repeating the experiment to see that as you dilute the catalase, the reaction rate slows down. Less bubbles form, or it takes longer for the bubbles to form, and you'll have a slower reaction rate. So if you have any questions, feel free to email your professor or email me and let me know what your questions are.